In the last chapter, we started looking at the way enzymes, through their structure, can confer function, especially to decrease the activation energy of certain reactions so that these are physiologically available inside of cells. And so a lot of what we saw is we were able to actually see, for instance, different intermediates along the pathway. So remember, if we were looking at a reaction coordinate, we might expect to see if this is the substrate and this is the products, we might see some sort of intermediate. And that turns into some sort of depiction of what the catalytic cycle is for an enzyme, such as it starts from substrate and it reacts with enzyme or binds to the enzyme to form some sort of enzyme substrate complex, which we'll denote as ES, and we'll see again very soon. And then this turns into some sort of intermediate, EI. So if that's intermediate, all right. And then that turns into a product. And then you close the cycle by releasing product. You have free enzyme. And then that's prepared to take another substrate and go around the catalytic cycle again. And so the question is then, how do we actually figure out what these intermediates are? How do we know what they look like? Because that's very critically important for trying to figure out, well, so remember the whole point of looking at these is to try to figure out structure-function relationships. That is, why does the 3D structure of the enzyme confer this activity? Why does it lower the activation barrier? And so how do we actually figure out what these intermediates are and therefore find out what's important about each protein that allows for these catalytic cycles to occur? Okay. And that's what we're going to discuss in the next few lectures, or the next few video lectures, about how we're able to actually start a study to figure out structure-function relationships in enzymes. How do we look for intermediates, and then how do we correlate structure-function relationships to these activities?